Okay, so now we're gonna move into more of the data analysis. So at this point, you have performed an experiment, sequenced it, and the Illumina software will produce these base calls that are just telling you what nucleotide is on each position of the sequence. That's the BCL file, and they have also software that converts that file into a human readable format, a FASTQ format, that you can actually, so that stores all the information about the sequences. So there's quite a lot of data pre-processing before you can start actually analyzing it. So we will go through those steps. I don't know how many of you have actually done any of this. If you ever receive the actual data, then you will have to perform all of these steps. It's not really feasible to do it in your personal computers. Depending on the programs you use, some of them might require quite a large amount of memory, and it is also many programs that need to be installed, so we are not gonna do it hands-on, but I'll try to do a demonstration if I can connect to the server. So usually in your institutes, you will have access to a cluster or a server where all of these programs should be already installed and available, if it's an institute that works with next generation sequencing data. So yes, the BCL files that have the base calls are binary files, so they need to be converted into FASTQ files. Again, there's a software that does this. In many cases, you will receive the FASTQ files directly, so this will be done for you by whoever performs the sequencing. And in that step of converting the BCL files to FASTQ files, the samples are demultiplexed if that's necessary. So remember, if you pull together different samples, each with a different barcode, then that barcode is read, and all the reads from the same barcode are put into a different file that then corresponds to the original sample that you sequenced. So at this point, you will have a file for each sample with all the sequences. So again, I'm not sure if all of you are familiar with FASTQ files and what are the different formats to store the data. Just really quickly, so FASTQ format is 4 lines for each sequence that you have. The first line is the header. It starts with an @ sign, and then it will have some sort of ID. The second line is the actual sequence of the read. Then there's a third line that just has a plus sign, and usually it's empty. Sometimes it has the ID of the read again, but that's optional. And then the last line is the actual quality of each of these base calls. So if you remember, we mentioned that when the software analyzes the images and decides which base was incorporated in that cycle, it will also assign a quality score of how confident it is that that base call is correct. And so that is the fourth line. And we'll see what all those characters mean in a little bit. So just on the header, sometimes you can store quite a lot of information there. So if you get a file that comes directly out of one of the Illumina machines, you will, for example, look more like this, and you have a lot of information there. So it will actually contain the ID of the instrument that was used, the ID of the run, the flow cell, the lane, the actual XY coordinates of which cluster it was being sequenced. So then you could actually trace that back. And if it was multiplex, it will have the sequence of the indexes that were used, so you can make sure that you have the right sample. But as I said, so the ID is anything you want to put in there.
Is that another line you said, or it's another type of file?
So this would be the same as this first line. It's just an example of how it might look. It might have whatever in there. Yeah. Is it helpful to have, is the pointer clearer than just the mouse? Yeah. Okay.
What would the third line usually be then for?
So the third line is usually empty.
And why does it exist?
Yeah, I'm not entirely sure. It might have had an initial use that then...
Maybe just to separate the sequence from the quality, like just to make sure that you know which is which.
So that you have this separate as well.
Yeah. Well, yeah, so it all means also that if you have a FASTQ file and you count the number of lines divided by 4, then you know the number of reads.
And it doesn't give you the strength or something.
No. It's always a plus sign.
It's absolutely followed by like any extra description on your sequence.
So you can store information in there.
You can, put again your sequence identifier and maybe further description.
Yeah, but generally it will be empty. Mm-hmm. So the quality scores, so again, they are indicating the probability of the base call being wrong. And it is called the Fred quality score. So this is something that, again, you'll encounter many times. So the quality score equals the -10 log 10 probability that the base call is wrong. So that means, for example, that if the quality is 30, there's a 1 in 1,000 chance that the quality is wrong, that the base call is wrong. Yeah.
How is it computed, the probability?
So that's, so base calling algorithm. So Illumina has developed its own base calling algorithm, and it will take into account like the intensity of the signal in the picture, and it will calculate noise. So it's a quite complex process. Yeah.
Is it based like sequence dependence, the base quality? Do they affect some differences depending on the base that are before?
I don't think so. So there are some, in terms of error patterns, so of the sequencing itself, there is some sequence dependence, but not on the base calling. That's on the sequencing itself. Yeah. Now, as you can see, you're not actually storing a number in there. So these quality scores have been converted to just a character based on the ASCII encoding. So the quality plus 64, and then whatever character corresponds to that value, that's what you actually have in the FASTQ file. So if you take the ASCII encoding of these characters, then that corresponds to the actual quality score. I see a lot of confused faces.
Which characters?
So in this line, so the first character is the score for this G.
For each base.
But this, if you convert this ampersand to the actual value in ASCII code, that might be 25.
So for each one of these characters, there is a different probability.
Yeah.
Yes. So that's it. That's for each base.
So how confident you are that that's related to you.
So the number of characters in the sequence must be identical to the number of characters in the quality line. Yeah.
Can I just add that there's some older FASTQ data types where it's a different offset, 33. So
Yes.
that's,
Okay.
nowadays it's 64, but occasionally.
Yeah. So if you analyze older data, then you might need to just be careful with what's the encoding there. Yeah. Okay. So once you have your FASTQ file with all the sequencing reads, there can be several quality issues, and you want to spot those as early as possible and just really ensure that the data that you're going to spend time analyzing is actually good quality and worth spending time on. So some of the most common issues is that you can have, for example, adapter contamination. You can have systematic failure at specific sample cycles. So for example, whilst you're sequencing, if there's a bubble that flows around the flow cell, that will interfere with actually the imaging, and so the quality of that particular cycle might be bad, and so that's something that you need to take into account. And something that is very common is that there is substantially lower quality at the end of the read. So so when you get to more and more cycles, then the quality starts decreasing. Can anyone think of why that might be? So why would Illumina sequencing get worse as the cycles progress? Okay, so let's say, if you remember, you have a cluster for each of your sequences, you've amplified it into a cluster, right? And you have many, many identical molecules. And then in each cycle, you will add one nucleotide. Now, as we said, reactions are not always perfect, so it might be that in one cycle, for whatever reason, a nucleotide is not added to one of the particular copies. So that sequence now is going to be lagging behind one nucleotide. So on the next cycle, Whereas all of them will incorporate the correct base, one of them will be one behind, and it will give you the wrong color, right? So if you have many copies of your sequence and one is wrong, it's still the majority of the signal will be correct. But as the sequencing proceeds, more of these errors will accumulate, and you will start getting a lot of more of these incorrect additions. And so it will get to a point where it's just too noisy and it just doesn't make sense. And that's why it has to be short reads, why it cannot just go on forever. Okay, so this is something that you will see. And again, if the quality of of the bases at the end of the read is just too low and too noisy, you might want to just get rid of that part of the read. Also, just in general, the library that you sequence might have, for example, low complexity. So that means that you didn't have that much starting material to start with. So you didn't have many different molecules. You've amplified that by PCR to a lot, and then you have a lot of PCR duplicates. So you're just seeing very few molecules a lot of times, and that's not really good quality data. So that's something that also you want to just spot from the beginning. So there's quite a lot of tools to QC your sequencing reads. Fast QC is one of the most popular. It's just, it will perform just most of the very basic QC steps. So you provide your FASTQ files, and then it will give you a report with different metrics. So we'll just go quickly through this. You might have seen these reports in the past. So this is the first, one of the first things to check is just the quality of all the bases per cycle. So on this one is a good quality sample, so you can see that most of the bases of the qualities are very good, and it's quite uniform across all the read. It starts getting a bit lower at the end, but it's still very good quality. And so again, all the values in this green area would be considered good, low error base calls. This is an example of a sequencing run that really failed. So you're only getting maybe 15 bases that are quite good, and then the quality gets really bad, and you cannot really trust that sequence. So if you see this, you wouldn't even bother analyzing that data.
Is this for one sequence, or is it
So this
for the whole run?
this is for all the sequences in that file.
This is for all the sequences?
Yeah, so that's why you have a
That's an average.
that's why you have a box plot.
Okay.
So that's the distribution across reads.
Why should that happen? Like, I never saw any run with bad quality, so I have no idea of what could create that.
It's because you're young.
Yeah.
So, so if you had analyzed the data when Illumina wasn't that good, this would happen more often. These days,
Yeah.
pretty good.
if this happens, then something really bad went really badly. It's not that common, but
Still happens.
yeah.
But I want to make sure I understand correctly. Is it like all the little parts, they are the same length?
So this is each cycle. So this is base 1, base 2, base 3, base 4.
Yeah.
And then it's taking just all the reads that you have and
Just
just
from one cluster?
from whatever it is in your FASTQ file.
Yeah, I mean, but if
So-
you have like the whole genome, for example,
Yeah.
the whole exome,
Yeah.
and it's from 1 to 40 here.
This would be if your read length was 40.
So, okay.
So
Okay.
it's a fixed read
Each
length,
time
yeah.
it's a
So it's for
That's
for
why
the
you
same
can-
position.
Okay.
So each
Yeah.
cycle of sequencing.
Okay.
Mhm. So yeah, again, if you analyze newer data, this will probably go to 100 or 150 for the length of your reads. Then this is just an average across the whole read. So again, just taking the average quality on all your read length, and so you want to see that most of them have a very high average quality. If you see, for example, a bimodal distribution here, so you might have some sequences that are good, but some sequences that are consistently bad, and you might want to filter those out and just get rid of them. Questions? No? Okay, so this is now again the sequence quality, but on the like the spatial location in the lane. So again, as we said, you have your lane. you will have the clusters located in different parts, and these will be scanned, and the photographs will be taken in chunks. And this is, again, the average quality, and blue means that it's good, so you want to see uniform things. So this is, again, to spot if something happened with a particular region of the flow cell where the sequencing failed. So in this case, for example, you see quite random or scattered regions that just have lower quality. This probably means that the clustering was too dense, so a lot of the clusters were overlapping, and so from all of those you're not getting consistent signal, and therefore the reads that you're getting are just nonsense, or a combination of the two. So from this file you probably won't be able to use that data at all. You can have situations like this one where you have just a particular region that just fails consistently throughout all cycles. So that might be just something happened in that particular region of the flow cell. It is often the edges where the imaging is not as easy, so the very end of the flow cell. Again, those sequences might be removed. It's also sometimes common to see that the quality drops at the end. So after a certain point, something happened and the sequencing failed after that point. So there was some blockage or something was just in the way of imaging that cluster. Some contaminant was stuck, and you cannot then sequence further. Again, this will be specific for just an area where that contaminant occurred. But again, all those sequences must be filtered out. Another thing to check is just the overall base content across your read. So you expect to have a fairly uniform GC or base composition that is consistent with the GC content of the genome. So it should be quite uniform across the whole read. If you have these patterns, then there's something odd going on where it's not random. And this is just, again, showing the overall GC content, and it should follow the distribution, the theoretical distribution of just your genome. It also reports the amount of N bases, so if the algorithm can just not make a decision of what's the base that should go in there, it might report an N instead. So again, if you have an increase of N's in particular cycles, something might have gone wrong with those cycles, and it's something that you should be aware of. If it happens at the end, you can just cut your reads to get rid of that. If it happens in the middle, it's a bit trickier. But if you're going to use that data, then it's something that you might need to consider if you're going to be doing base calling and just looking for mutations, then those parts of the read will not really be informative. The other thing to look at is how unique are your sequences. So as we were saying, if you have too many PCR duplicates, then what you sequence is not going to be unique sequences. So in this case, again, you have the sequence duplication level on the x-axis. So most of the sequences have just one copy, and then a few have two, but most of them are not in many more copies than that. In this case, instead, you have a much lower percentage of sequences that are unique, and then quite a lot that are just the same exact sequence many times. And that might be just something that got amplified too much, or you might be actually sequencing just adapter dimers instead of an actual DNA molecule, and then it's just the same sequence over and over again. And again, that is something that you don't want to analyze. It's just noise. And it will actually report any sequences that it finds that are overrepresented, and it will try to see if those correspond to the adapters, the most commonly used adapters. So it will tell you, for example, if they match the Illumina primers, that then you know that, so where those sequences are coming from. It might, so again, you need to remember if you have so you will fragment your DNA or RNA into hundreds of base pairs. But if you, for example, get fragments that are shorter than your read length, then you will read into the alignment right? So if you have a DNA fragment that is only 70 base pairs long, and you do 100 cycles of sequencing, you will read all the 70 base pairs of your DNA fragment, and then you will start reading the adapter that is ligated to it. So again, all those adapters need to be removed because it's not really part of your sequence. Now, RNASeq specifically has a few different particularities. So in terms of the sequence content, we've said that you should have quite uniform base composition along the read. But it's now been observed, and it's well documented, and there's a paper in there that studied this, that the first around 13 nucleotides are not actually random, and this comes from those hexamers that are used to do the reverse transcription. So if you remember, you just use what is called random hexamer sequences. They're not completely random, and they actually introduce these biases in sequence composition. So this is actually completely normal in RNA-seq. FastQC will report it as a fail in the quality, but that's actually a good sequence. So as long as after the first 13 to 15 nucleotides it stabilizes, then that's normal.
But the first part you have to remove anyway.
No,
No?
no, it's correct. It's just that it's not.
But it was a random primer added.
So it just means that it will insert or it will prime from a specific,
Okay,
more
that is
specific
part of the
locations. So you won't have just a random composition because it will be biased to specific
Okay.
sequences.
So that depends. So you can have random primers or you can have poly.
Yeah,
Yeah.
Okay.
Mhm.
What's more common nowadays? Is that?
I think both are used.
And random primers are if you're not looking for mRNA.
It's both. So you can do that with mRNA as well.
But only you like the targeted or like the poly are only for mRNA instead.
Yes.
Okay.
Yes.
Yeah. So if you do ribose depletion and then you sequence, you use poly A, poly DT, then you will still lose
Yeah.
all the non-polyadenylated.
Also, just coming back to the DNA, like for the fragmentation, do you
Mhm.
see some kind of patterns in this
Mhm.
cycle? Like, is it random or there is still a bias for it?
There is a little bit of a preference for the enzyme to insert in certain context. So it's not completely random.
Right.
It's almost, but it does have a little bit of a bias. Yeah. And again, in the report of what are the, so you will look for overrepresented sequences and also just k-mers. So you will see again in the first few cycles, you will have these k-mers overrepresented just because of the bias, but you will also, if it's RNA-seq, you will expect to sequence poly A tails at some point. So you will see these poly T and poly A sequences at the start or beginning, at the beginning or the end of your read. Again, that's normal. That's just, yeah. Okay, so again, most of the time you will get data that is very good quality. These days, Illumina sequencing, it performs very well, and so most of the time, all of this is just a quick check that nothing went horribly wrong, but it will usually be good.
Excuse me.
Yeah.
So what if this is based on all the sequencing point of view? What if there is a problem with the library preparation and still it is sequenced? Is there a way that we can find the difference between that?
Yeah,
Because
so
there might be a library contamination or library just is not very good and still it gets sequenced, but the sequencing quality that will come out of it will be good.
yes. Yeah, so usually if your library is poor, it will sequence not as good. So if you, for example, multiplex several libraries and one of them is of bad quality, you will get a lot less reads from that sample. So that's a first flag that sample wasn't very good to start with.
But do we know how many reads it will get?
So depends how many samples you put in per lane.
Okay, so in multiple samples, rest of the samples will perform good except for the first one sample or two samples which have a bad library.
Yeah. And in some cases, so again, if your library was quite bad to start with, usually these libraries are low complexity, so you will have a lot higher number of duplicated sequences. So not most of them will be unique. Again, that's telling you that you didn't have enough diversity in the RNA to start with. And then if it's more specific things, like you might spot them once you align the data, but we'll get there. Any other questions on just the QC of the reads?
If I understand correctly, the left one is from random priming and the right one is from poly-A priming, or not?
They're both from random primer. Priming is just different metrics. So this
how
is
it possible that you don't have this? Oh yeah, that's
you do
different
get it.
metrics. Yeah, okay.
Yeah.
Just for
Yeah, there's
RNA,
so many bases. Yeah.
once you get your fragment, do you still TA at the end to get oh no, what did I say? It's wrong. No.
for the adapter.
Yeah, I was
Yeah.
thinking for the adapter, but you...
Depends how you can. Like if you didn't add it during the reverse transcription, then you just ligate them with that method. Yeah,
But yeah, normally now it's during the reverse transcription that you do it.
it's more common. Yeah.
Are silencing RNAs and small RNAs and all of those types of RNAs? So can you neglect that and use poly instead of random, like the hexamers?
Depends on your question,
Depends what you.
right? If you're just interested in like mRNAs,
DNA.
then yes. I think so you just need to keep it consistent, right? If you're gonna compare two things, just use the same method. But yeah, you won't get any information about any other classes of RNAs. Yeah. And I guess we should mention also that so you can sequence small RNAs, so microRNAs, or those types of RNAs will need a specific library prep procedure, because of course they are very short, and usually we are filtering for things that are a few hundred base pairs long, which is not the case for those. So that's a specific protocol, but we won't go into that. Okay, so you've QC'd your reads, you're happy that they're good quality. The first step is to align them to reference genome. So you will hopefully be working with model organisms, so human or mouse, that have very good quality reference genomes, and then it's quite straightforward. If you're working with something that doesn't have a good reference genome, then that's a whole new challenge. And again, we won't go into the details of that. But yeah, so we're assuming that you have a good reference genome. And basically what you want to now know is all those sequencing rates, where are they coming from? So where in the genome they were generated. So that's the main task. Now, there's quite a few challenges, and a lot of them deal with actually the amount of data that we have. So we now, from an Illumina experiment, you can get millions of short reads. And of course, the genomes are also very long. So you have a large space to search to find these matches. So you can imagine that then it can become a really big task to actually align each of these reads. And then also, you know that there will be sequencing errors. There might be variation between your samples and the reference genome, and so the alignments can have mismatches or gaps, and you need to be able to allow that. So this is just a way to represent an alignment, for example. So at the top you have the reference genome, and then these are all the sequencing reads. If the base matches the reference, then it's just a comma. And then if it's a different base, then you have the actual base. So you can see that you will have random errors. Those are probably just PCR or sequencing errors. And then you might have a SNP of a base that it's just different in your sample to the reference, and then that's found in all of your reads. So the alignment needs to be able to identify that this is the location where those reads are coming from, but it's not a perfect match. So it needs to just be flexible enough to allow for that. And it might need to allow for also insertions and deletions. So very quickly, it gets into a quite complicated problem of just dealing with a lot of data and having to consider quite a few different scenarios. So there's been a lot of work done into actually producing alignment programs that are capable of doing this. And there's two main strategies that are used to make this task feasible. So first of all, there's a filtering step where you just want to very quickly exclude all the parts in the genome that cannot produce your read. So for this, usually the algorithms use seeds, so that's just a small region of the read that then you can just search for a perfect match in the genome, and just whenever there is not a match, all that part of the genome is just excluded. You don't even focus on that. And then to be able to do those searches in a very quick and efficient way, there's indexing methods. So again, if you're thinking that you can actually use the analogy of just if you have a book and you want to find a particular concept, if you have to go through all the pages, it's going to take you forever. So you go to the index, you find your concept, it tells you where in the book that concept is covered. So it's the same. In this case, you will have an index of the genome. Sometimes it's the reference reads, the sequencing reads, but most often it's the genome. So wherever, just all the different seeds that you could have, where in the genome they are located. And this is just commonly done using hash tables, which are just very efficient at retrieving these locations. Or there's suffix trees. So again, you just have all the different stretches of sequences that you could have in a tree structure that it's just very efficient at traveling that tree and finding the matches. And then there's combinations of doing indices with what it's called a Burroughs-Wheeler transform, which again, we won't look into the details, but if you're interested, it's worth looking into it. So the general process of doing the alignment is that you will get these seeds from your reads. So that's, again, just a short sequence. It might be 10, 12 different nucleotides. And then you will just look for that in your index. So where is that seed in the genome? And those are all your candidate regions where your read might map. And so now you will then take all of these regions and actually perform a complete alignment of the read. And that is the slow step. So hopefully you've now reduced that to only a few candidate regions, and then you can see if you can actually extend to a full alignment of the read that it's good enough quality. And then similar to the base calling, there's also a mapping quality, which again is just telling you what is the confidence that that position is the actual true location of the read. So from all the alignments that it finds, how confident it is that the one that is reporting is the true position. And so if you find that the read matches in two locations, then that mapping quality will be quite low because you cannot differentiate between the two. And the encoding is the same. So again, it's -10, the log 10 probability that the alignment is wrong. So again, a quality of 30 is 1 in 1,000 chance that the alignment is wrong. And so again, you wanna just take that into account. If you have alignments of poor quality, you might just wanna ignore those because you are not really certain of that information is correct. Okay, questions?
Do the alignment algorithms consider the quality from the FASTQ files somehow, or is it just for the?
Most of them do. So there's a lot of them, but yeah, most of them will have like scoring for whenever there's a match or an indel. There will be some penalty. If the base is low quality, that penalty might be less because then it's really likely that it's a sequencing error and the alignment is correct. So yeah.
So it is incorporated in calculating this probability?
Yeah, so for each alignment you will have also an alignment score that just tells you how good the alignment it is. So if it's a perfect alignment, then it will have a very good quality score. If it's an alignment with a lot of mismatches, then it will have a lower score. So just similar to BLAST, right? When you do a BLAST, and you will get that E-value that tells you just a similar concept. Yeah.
Do you choose your seeds, and what happens if there's a mutation in the seed? And then you don't have a perfect match to the reference, do you?
Yeah, so usually most algorithms will take several seeds.
One read.
Yeah. So one of the ideas is, for example, you will always have one parameter is how many mismatches you allow. So if you say, I'm gonna allow only K mismatches, then you divide your read into K plus 1 seeds. That means that at least one of them must be a perfect match. Mhm. Yeah.
Regarding what you are saying about penalties,
Mhm.
I mean, in those, there are different penalties associated with an insertion, a deletion. You have like editing distance,
Mhm.
and you can apply like Sankoff algorithm, or perhaps in the case of sophistry, like it's like Hoffman codes. Hoffman, is that right?
I mean, there's quite a lot of different aligners, and they all use different methods and different scoring procedures. So yeah, there's a great diversity. So different aligners will use different methods, just both for mapping and for scoring the alignments. So it will be important once you choose which aligner you're going to use, just to read the manual and just work out how they actually do these things. So you will have a lot of parameters of how many mismatches you allow or how low the quality can drop, depending on what the aligner does internally. Yeah.
A question about secondary alignments.
Mm-hmm.
Is it all calculated at the same time, or is it first like it finds the first alignment and then it looks if there is something because of the lower score or something?
Yeah, so again, different aligners do it differently, but usually you will basically have several or all the possible candidates from the seeds, and then you will try to look if each of them is a valid alignment. So you will start extending it, and for example, some aligners will say, well, if the alignment score drops to a certain point, then I don't even bother. That's not a valid alignment, and forget about it. There will be cases where you have, especially again for repetitive parts of the genome. So in this case, you have a repeat here that it's just one base difference. So it's a G here and a T there, but the rest of the sequence is identical. You have in this case two reads, so that's a read that matches perfectly here, and then it matches with one mismap there. And if you're allowing mismatches, then both alignments would be valid. In this case, again, it aligns perfectly there and then with one mismatch here, but this lowercase g indicates that that is a bad quality base. And so you are really not sure where it comes from, because it might, even though it matches there perfectly, because that base is low quality, it might be just from either of them. So you wouldn't be confident in calling one over the other. But for any of these alignments, even if it has one match that is better than the other, because the other one is quite close, your mapping quality will be lower, because your confidence is just not that strong. And so, again, each aligner will have different rules onto what they do with this. Some will report only the best ones, some will report all of them, all the different possible alignments, some will report a random set. So again, depending on the program you use, you just need to make sure that you are aware of what exactly it's given you and how it handles these type of cases. So, as we were mentioning from the DNA sequencing side, in this case, again, having paired and reads helps in the alignment step, especially for repetitive regions. So if you have a paired, so if you have sequenced both the ends of your DNA fragment, RNA, even though one of the reads is completely mapped to a repeat and so has a location both in this repeat and in the other, if the pair aligns to a unique part of the region, then you can assign that pair to a unique place. And so all these reads will now become uniquely mapped, whereas only the ones that are completely contained within the repeat are mapped to all the different locations. So if you have access to paired data, usually you have better mapping accuracy than compared to single end data. And that's, again, true for read length. So the longer you read, then the more unique hits you will get, because then you have higher chances of actually spanning the repeat into some unique sequence. another thing to consider when you're doing an alignment, again, usually you will use the reference genome, but if you have your samples are from substantially different genome, then there's going to be a lot of variation, and that is just going to make the quality of the alignments a little bit worse. So for example, the reference mouse genome is from a particular strain, so the black 6 mice, but there's many different laboratory strains of mice that might be used for different experiments. So if you have a cancer model, that might be a particular genetic background that is different to your reference. There are now, for example, projects that have sequenced like 20 or 25 different strains of mice. And so all the variants that are different to the reference are available. So you might want to consider to actually impute those positions into the reference to have a more close reference. Or in the best cases, if you're working with mice, actually they have created assemblies for each of the strains. So instead of mapping just to the reference, you might want to actually take the correct genome. Yeah, and again, if you're working with a model or with an organism that doesn't have a genome, you might need to use the closest genome available, but then you're going to have a lot lower mapping results. So there are many different mappers available. So this is just a timeline of like the first mappers were developed when next generation sequencing became popular, and you can see that when it became like very like the explosion of next generation sequencing, there was a lot of programs that were developed. Now it's slowing down again because basically we have explored most of the possibilities of how to make the alignments better. And so this is now considered pretty much a solved problem, though now that we have longer reads, that will become again a new challenge. Anyway, there's a lot of programs that you can choose from, and they will have all different methods in terms of how they find the alignments, how they score, how good the alignments they are, how quick they are, how much memory they need, and so on. And there's some that are more popular and that have been used now for a lot of years. So some of the most popular are here, and they are based on different indexing and search criteria. Bautai and BWA, I think, are some of the most widely used, and again, you might just get some data from these aligners. But there is really no best aligner. If you read the papers of any aligner, they will say that they are better than everyone else, but it is quite specific to the type of data that you have. Some perform better with certain types of data than others. And so you really just maybe want to try a few and compare the results and get a feel for what works best with your data. And then, as always, just keep things consistent. So don't change aligner halfway through. Just keep the parameters the same. And I do think it's really important when you are going to perform this analysis for the first time that you get familiarized with what the aligner is doing and how it's doing it. They all have a lot of different parameters that you can change and you might want to adapt for your specific samples. So that's something that it's worth just investing a little bit of time in, and just to actually understand what comes out of it, how it achieved those results.
So how long does it take at the moment, like on average, with average equipment?
Depends a lot on the program you use.
The quality of the data, let's say, like 30x. So how? Depends
a lot on the aligner that you use. There's some that are very quick but need a lot of memory. There's some that don't need so much memory but that then take a lot longer.
But it's like in minutes, hours, or days?
It's usually in between minutes and hours. But it can take days depending on what you're using and how much data you have. Yeah. And most of these, or a lot of these programs now are parallelized. So if you have access to a cluster where you can use several CPUs, then you can parallelize and everything becomes a lot quicker. So if you have access to 18 cores at the same time, then it will take a lot less than if you have to do it in just serial mode. So we now there are some formats that have been agreed and that are widely used. So each of these aligners might have their own format on how they report the alignments, and then that's a problem because depending on the aligner, you need to consider different things. So now there's standard formats that most people use, and so for the alignments, that's the SAM format, or the sequence alignment and map format. And so that's just a tab delimited text file, and it has certain sections and very well defined requirements. So it always starts with a header section, and the header lines start again with an @ sign, and all the values or all the information is stored by having a tag that will tell you what sort of information it is, and then the actual value of what So the first line is the header line, so it will be indicated by HD, and this usually has like the version of the SAM format. It can have information of whether your alignments have been sorted by the position on the genome or not. Then you have the sequencing dictionary, so this is the SQ lines. So you will just have information of what genome you have been mapped to. So it will have the sequence name and the length of that sequence. So one entry for each chromosome that you have used. There's RG lines, so that's the read group. So again, if you, for example, had multiplexed samples and then you have a barcode identifying the sample, that will be stored in these lines. And then there's optional values that can go there, so you can store information like the sequencing center, when it was sequenced, the actual platform, the insert size. There's quite a lot of space there for all sorts of information. Then there's the PG lines, which are actually programs that have been used. So these will usually have the program name and the version, and many times the actual command line that was used. So that's quite useful to just have, to trace back how that file was generated. And then there's optional comment lines, which most of the time are empty, but if you want to add a comment, then that's how you do it. So this is how it looks like. So again, you'll have the header line, that's the version, and then it's telling you that it is sorted by coordinate. This is the dictionary, so chromosome 1, and that's the length of chromosome 1. So these are all the names of the chromosomes, so those will be scaffolds that are not placed in the genome. The RGs, again, so it's telling you that it's Illumina data, and then that's actually like the sample name, and that's the sequencing center, for example. And this is the program. So this was mapped with PWA, right? So, and then there's the version that was used, and then there's the command that was run. So that was the reference genome, and then that's the sample that is being mapped. And in this particular example, this sample has also another program line. That means that there's another program that has been run with it. In this case, mark duplicates. So this is again to mark PCR duplicates. So if this was DNA sequencing, you would take all the alignments that start at the same exact position. They are flagged as PCR duplicates, and you can choose to remove them or not. And so that's again, you have the command line that has been used in there. So again, this is all just general information of what has been done to the FASTQ file to get to that alignment file, and the details of the programs that have been used. So after that, then you have all the alignments, and again, you have 11 mandatory fields. So you will... Yeah.
When you go from the duplicates, can you also remove that at a later step? Or is there always... So this duplicate marking, they're still in the BAM file, are they? Can you also do that then at a later stage, removing them, or
so
this is just marking them basically as duplicates probably?
You have the option. So in the command, you can either say just mark them or remove them.
Okay.
So it's up to you.
You can also do that later. They're still in there basically.
If you chose not to remove them, yeah. So whenever you run it, you say whether or not you want to keep them.
So they have to run it again to remove them? Oh.
Yeah.
Well, you can run it from the beginning, specify to remove them.
If you tag them, then you just can also filter with the tag and then you
Yeah. Depends what you're gonna do. Yeah. So again, for RNA-seq, you normally won't do this because they are very likely not PCR duplicates, and so you wouldn't remove them because then you are just losing information that is actually valuable. For DNA sequencing or things like ChIP-seq or ATAC-seq, you might. Okay, so the alignments, again, you will have one line per alignment and 11 columns. So first is the query name, so that's just the ID of your read, and then a flag that it basically gives you information about how, like the characteristics of the alignment. So this is again just set criteria, so it will tell you, for example, if your alignment is unique or not, if it's unmapped, if when you have like paired and data, if you're looking at the first segment or if it's a secondary alignment, all of these things. So depending on the value that you have in the flag, then it's a combination of all these characteristics, and then you can get information from that. Then you will have the reference name, so where it is mapping, so this will be the chromosome, and the actual position where the alignment starts. And then the mapping quality, so what we were discussing of how confident the aligner was that this alignment is correct. Then you have this cigar string, which gives you information about the alignment itself. So it will use different letters for what's happening. So an M means that it's a match, an I means that it's an insertion, a D means it's a deletion, and so on. So if the read mapped perfectly and it's 150 base pair read, then you have 150 M, which means it was 150 matches. If you had an insertion at some point, then you might have 100 matches, 2 insertions, and then 48 matches again. So it just gives you an idea of actually what happened along the read. Again, if it's paired and sequencing, then you will have information about the mate pair, so where the mate pair is mapping in the reference, so the chromosome and the position, and the insert length that you would get from those two alignments. And then you get the actual sequence of the read and the quality. So these should be the same as you had in the FASTQ file. So as I said, these 11 columns have to always be present. If the information is not available, then it can have a 0 or an asterisk. So if it's paired and data, those columns will not have a value, but they should still exist as columns. And then there are optional fields that each program uses their own optional fields to just attach added information. So they might have, for example, how many mismatches there were, how many additional alignments it found. Again, you'll need to go into the documentation of the particular aligner that you're using, and then they should document what sort of information they're giving for each alignment. So an example, so that this is a bit clearer, this is one alignment line. So this is the name of our read. Again, that's the flag. So in this particular case, this 99 is telling us that the read is paired, that it's mapped in a proper pair, this was paired and data, that the mate is in the reverse strand, and then this one is the first in the pair. So it's mapping to chromosome 10 in that particular position. That's the mapping quality, so this is a very confident assignment. And that's the cigar string. So in this case, we have 44 matches, and then 106 soft clipped. So that means that that cannot be aligned anywhere, so it's just not aligned. So it only matched the first. So that could be adapter, that could be a poly A tail, or just low quality sequence that could not be aligned. Then that's the information about the mate pair, the sequence, the actual sequence, and the quality. And then these are the optional fields, which will be specific to whatever program you're using, right? Okay, and so this would be the text file, so that's SAM files. These can be quite long, large files. If you have millions of reads, you will have millions of alignments, and those files very quickly become very big. So there's been a lot of effort in creating compressed files that take a lot less space. So BAM files is the binary compressed version of a SAM file. So it will be a lot smaller, but you cannot read it directly because it's binary. But also it can be indexed. So the similar concept that we were using for aligning. So if you just want to retrieve the reads in a specific region of the genome, if you have an index, then you can do that very, very quickly. Of course, that will need that your alignments are sorted by position so that this works. Again, there's the convention that if you have a BAM file, the index will just be the name of the BAM file plus BAI at the end. So these are all just conventions that everyone follows. And now more recently, in the past couple of years, there's been the development of CRUM files, which are even more compressed than BAM files, so they can be even half the size of a BAM file. And they achieve this by basically doing the compression in a reference-based manner. So what this means is that you assume you know the reference, and so you only need to store what is different compared to that reference. So you don't need to store the whole alignment, you just need to store the bases that don't match, right? And so then you can save a lot of space. But that means that you need to have the exact same reference that was used for mapping to be able to reconstruct the reads, the alignments. The manipulating these SAM, BAM, or CRUM files usually is done with some tools. So again, these will be tools that allow you to convert from one format to the other. They allow you to merge different files, to do the indexing, to sort either by position or by read name. And again, all of these tools are well documented, and there is an interface to use it from R, and sometimes that's convenient. And another set of tools that is quite useful is Picard tools. So the marking duplicates, they have a quite good tool to find the duplicates and mark them. That's something that you might encounter, and they have a lot of different tools, like to collect metrics and do different things to the files. Okay, so as we said, you cannot perform the alignments yourselves in your computers, but we can have a quick demonstration of how this would be done if we were actually doing this. So hopefully the connection will be good enough. So is that... Can you actually see, or is that too small?
It looks like the way you see it's blue.
In white it's okay.
So the important is the white.
Is what?
So ignore the blue, it's just my directory.
Don't we get the inferred genome?
Say again?
Take it.
Do we get the inferred genomes from all the alignments?
So you mean to assemble it?
Yeah.
You could try. So if it's short reads, you will usually get relatively small chunks, and you will then just hit repeats, and then your alignment will be broken.
Yeah.
But yeah, there's a lot of work done on that, and usually you have to combine the short reads with something longer.
That's a good application. Yeah.
One question.
Yeah.
The compressed files are lossless? I mean, from the compressed file you can reconstruct...
As long as you have the right reference.
All right.
Yeah. So from the BAM you can, and from the CRUM you need the reference.
Yeah.
Okay, so in here I've just created a dummy set of only like 50,000 reads, which of course is not much, but just so that everything is quick. So this is perdent data, and you will have two files, so one for the forward reads and one for the reverse reads, and they are all in the same order. So if we look, So again, as we discussed, this is a FASTQ file, so you will have the header, then your sequence, empty line, and the qualities. And then if you look at the other file, actually, so they are paired, right? So this read is exactly the same one in the other file. So they are all in the same order. So you know which one corresponds to which. And then, so we have reference information. So in this case, it's, why is it doing that? Okay. This is mouse data, so we use the mouse reference genome. So this is just a FASTA file. So again, you will have a header line that is just starts with this greater than sign, and then the name of the chromosome, and then the actual sequence. And so these will be just big files with all the sequences for all the different chromosomes. If you were to do, so you will first perform your FastQC and just look at your reads. If you're happy with that, then you might perform the alignment. So, for example, we can use BWA to do this, and then when it will give you like all the options of what you have to do. So you can provide different options on the specific, like how many matches you're going to allow and so on, and then you give it the reference genome, the index of the reference genome, and then you're either single end or paired end FASTQ reads. So in this case, we will just do PWA mem, so that's the program. We're gonna ask it to save the output in this folder, BWA, with that name, and then this is the reference that we wanna use, and then our two sequencing files. So we do that, and because it's only 50,000 reads, it will be quite quick, I hope.
Do you use for reference for, I mean, how much, but for units, do you also work on units? What kind of gene do you use there? Is it like the 38 or the 37?
The 38, so it's always recommendable to use the latest assembly because it's the best. Yeah, and if whenever you're working with data that has been already processed, that's something to keep in mind. There's different assemblies and you need to know which one because the positions will change from one assembly to the next, right? So this, as you can see, is the version 38, which is the most recent one. So that's done, okay? And so now if we look at what happened in BWL, now we have our sum output in there. And if we look at it, so we have the header. So again, this is the sequencing, the sequences that things are being mapped to. So chromosome 1, and that's the length, and so on. And then we have the program line, okay? So there's the command that we just run, yeah? And then these are the actual alignments. So again, just telling us this read maps to chromosome 11 in that position. All 150 bases are matches, and then the mate pair maps to the same chromosome, so that's the equal sign at that position, and that gives you an insert size of 191 base pairs. Then you get, again, the sequence and its qualities, and then some information. So these will be, for example, the number of mismatches for that alignment. And it's just like that, right? Now, if you look at these, so this file is 49 megabases. So again, it's not very big because it's only 50,000 reads, but you can use now some tools to convert this to a BAM file. So some tools view, again, it gives you all the options of how to use it. So with the -b, it will output at BAM instead of SAM file. -h will include the header.
Can you reuse the window?
Is that better?
No, we don't see the
Oh,
beginning of
sorry.
the screen. Oh.
Yeah, that wasn't useful. Yeah. Okay, so yeah, these are the options. So in this case, we want to do some tools view, and we want to output in BAM, and we want to keep the header. So we give it the input file, and we just get that into now a BAM file. right? So that will be very quick. And now you can see that it's a lot smaller compared to the SAM file. We can also sort this file so that it's sorted by position. So again, if you just type the command, it will tell you how to do it, and then you could index and then retrieve the sequences from a very particular region of the genome. Okay, so
Sorry, can you say what was the sort again for?
to have all your reads aligned by the position in the genome.
Is that then to have the BAI file, or do you set something different?
No.
Index sorting? Is that
So to be able to convert to build an index, you need to have your alignment sorted by position.
Okay, so this is something
Yeah.
you do first, and then.
Yes, so you would do some tools sort of your BAM file. Sample 1 sort BAM. And now you can do some tools index of this one. And then that has created this BAI file. So that's just the index. But now I can tell to some tools view. So if I give it the sorted BAM file.
One question, why is it now smaller, the sorted than the sorted? I mean, does the sorting also do some compression on it?
Yes.
Okay.
Yeah. So when you have all the alignments that are mapping to the same region all together, then you can further compress that. You can save information on that as well. So now you can say I want just, I don't know, chromosome 1, position 3 million to, yeah, I mean, I don't have, it's like that. But because this is such a small file, I don't know where the reads are, and just, yeah. But then it will just retrieve them very quickly. Okay, so we can go now into what's specific about RNA-seq data. So if now you're thinking you have actually sequenced RNA and you have your reads, what would be the issues that you would have when you're aligning them to a reference genome? So what can you think that would be different?
DNA to RNA, you mean, or?
Yeah, so you've converted your RNA to DNA and sequenced it. Now?
Splicing and stuff like that?
Yes, exactly. So now in the RNA you have all the exons now are stitched together and you're missing the introns. So now if you just take the read, of course that doesn't map at one location in the genome because it's just fragments that are scattered around with big gaps, and introns can be very big sometimes. So the aligners that were developed for DNA-seq data are not very good at actually mapping RNA-seq data exactly because of these reasons. So now you need actually to be able to take basically regions of the read and allow them to have big gaps between them. And so there's two options on how to deal with this. So you could map the reads to the transcriptome instead of the genome. So if you just take all the genes and actually remove all the introns and just create, instead of chromosomes, now you have transcripts and then you map to that. Or you modify your program so that you can allow these big gaps in the alignments. So let's look at both of them. So in the first option, if you map to the transcriptome instead of the genome, so again, now your reference is just thousands of transcripts with just the sequence of all the exons pasted together. So let's assume you have two different isoforms of the same gene. One has 3 exons, and the second isoform skips exon 2 and just has 1 and 3 together. So all the reads that map to exon 1 and 3 will align to both isoforms, and those that are in exon 2 will be specific to that isoform. So that means that immediately all the reads that have exons that are shared between isoforms will be mapping to multiple reference sequences. And so you will need a different approach to then consolidate those in a sensible way. So it's not the easiest approach. And also the big limitation is that this will work if you're working again with a very good annotated genome where all the genes have already been defined, and then you can actually trust the annotation. But also no genome is perfect, and one of the strengths of RNAseq is that you can actually discover new isoforms and completely novel genes, and that's something that you would lose if you're just basing it on the annotation. So perhaps more appealing is to actually modify your algorithm to be able to allow the reads to have these big gaps that span the introns. So now we want to have some reads will map perfectly to the exons, and those are not a problem, but some reads will start in one exon, then they need to be interrupted and continue in a segment that is far away in the genome. So again, in going back to this graph, all those that are in red are specifically for RNA-seq and are able to deal with splice junctions. And again, they differ in their memory requirements, in their speed, in their different ways of how they solve this problem. Some of the most common ones, and that you might have heard, are top hat and star, but there are many of them. So we'll just talk about those two as examples of how they deal with the splicing. So top hat was one of the first ones that was developed, and probably that's also why it became so popular and has been maintained up to now. So it does the alignment in 2 stages. So first of all, it just takes the reference and all the reads and just maps them with bow ties. So that's a DNA-seq aligner, so it will just try to find contiguous alignments. And a lot of the reads will map because they do not span exon junctions. Yeah. So you will have a certain proportion of reads that are mapped, and then a certain proportion that are unmapped, and those are set aside for now. Now, with the ones that have been mapped, then you can just get a consensus. So all of these regions will be joined together into this fragment, and so these will be our putative exons. So we assume that these regions correspond to what will be exons. And then you can look at the sequences around them and identify the possible splice junctions. So again, you always have a splice donor and a splice acceptor sites that are defined specific sequences. So you can actually make a prediction of where the splice junction is going to be and create now a reference that has just these splice junction sites. So you now generate, instead of mapping to the reference, you're going to map to the exon junctions. Yeah. So that's the second step. So you recover all the unmapped reads, and now you're mapping them to the splice junctions, and just to see which ones are compatible with that. And then you put both sets of alignments together, and so you should have both the contiguous and then the spliced reads. And then a few years ago, there was a newer version released of Top Hat that now can deal also with like indels and deletions, and it's meant to be better. So yeah, that's one option. The other program that has become a lot popular in the last few years, and that was used for the ENCODE project, is STAR. The difference with Top Hat is mainly that it is a lot quicker, so it's very, very fast, but it requires a lot of memory. So usually you will need about 35 gigs of RAM to run it, so you cannot run it in your personal computer. But if you have access to a cluster, then it's very efficient at doing it. And so it uses a slightly different approach. It doesn't rely on a DNA mapper to start with. It just takes all the reads and it finds the well, it's called the maximal mappable prefix. So it will take all the sequences that it can find as exact matches in the genome, and whenever that exact match finishes, then it cuts there and then it starts again. And so if there was a splice junction here, then it can map this to one region and then start again from there and then find it in a completely different position. Mm-hmm. And then for each of these, it will try to extend, now allowing mismatches. And it also allows you to have, again, the end of the read might not be mapped. For example, if you're reading into the poly-A tail, you have to allow for that sequence to just basically not be mapped. And then again, so it will just take all of these MMPs and stitch them together and then create a full alignment and score them just like all other alignments do, and just give you the best possible alignment. Again, there's no best alignment. If you compare the results from Top Hat and Star, they are quite similar. You will get very similar results. Each of them will have certain biases. So as long as you keep it consistent and you always use the same parameters, then you should be fine. So let's now, instead of doing our alignment like we did with BWA, we will use Star in the same data. So now this is the command. So again, we're just giving it a reference genome and then our reads and a few different options. So in this case, we're asking that the output is in BAM format and it's sorted by coordinate. You can in Star actually ask it to quantify. So if you give it a gene annotation, then it will, as it maps, also count the number of reads that map to each gene, for example. So that's quite convenient for RNA-seq data. And this will take a little bit longer. The slowest part is loading the genome, just because it's large, but once that's done, then it's quite quick. So any questions up to here? So it's RNA-seq data in this case. Just
So
50,000
it's the same data, and before you used it as DNA and everything when...
Well, you can use BWA to map RNA-seq data. It's not going to work very well, but
just as a showcase.
just yeah. So that's done. It took a minute or so, and now we have. So it creates a BAM file with all the results. It will give you some statistics. So if you look at this file now, it will tell you, for example, like the number of input reads, the average length, how many mapped uniquely, how many were multi-mapped, how many were unmapped. So things that it's useful to just look at. Again, just how the percentage of reads that map uniquely already tells you how good your sample and your library is. And then if you ask for the quantification, then it will give you also this reads per gene file where you will have all your gene IDs. So in this case, I gave it a ensemble annotation file. So these are ensemble gene IDs, and then it's telling you how many reads are mapped to each. This will be mostly 0 because it's only 50,000 reads, but yeah. But just as an example, if you take these BAM files, IGV is it's a program where you can actually visualize data. It's quite useful. If I go full screen.
What is this program called again?
So it's IGV, so it's the Integrative Genome Viewer. And so you can load BAM files and all sorts of annotation files in it. So usually you have your genome here, so you could choose to use human or mouse and the different versions. So in this case, we're using mouse.
Sorry, we keep having this problem.
Sorry.
Also for the slides, it's similar. So I mean, we are able to read, but then just somehow when you go full screen mode,
It doesn't project the whole thing.
Yeah, it doesn't work.
Okay.
on the left-hand side, we also
I
have.
think it's the matter of projector.
Yeah.
But you have set the resolution of the benchmark.
Okay.
Yeah, some things are wrong.
So I think that's the full screen. Right. So you have here it's the genome. So you can choose from already available genomes, or you can load your own genome. And if it's one of their genomes, it will also have the annotation. So these are RNASeq genes, for example, and then again the blue boxes are exons and connected by lines which represent the introns. And it has all sorts of information about its location and IDs and so on. So in here I've loaded the output from the BWA alignment, and so you can see that now we have reads that are aligning in this region. So each one of these, you can zoom in, each one of them is an alignment, and then if you have mismatches, then it's just colored by the base. Yeah, so that would mean that base, instead of being the reference, is actually, I think, a G. Yeah, so in here you have the actual sequence. If you zoom in enough, then you can actually see the sequence of that, and it's color coded.
What kind of data is it? Is it bulk?
This is bulk, yes. Okay, and again, you can see all the information that you have stored in the BAM file, then it displays. So you have the read name, the read length, mapping quality, the cigar string, all of these things. So it's quite useful sometimes just to double check that your alignments were okay, that there's no weird artifacts going on, and so on. But as you can see, we're not recovering that much of this gene, so we're only mapping the end of it, really, and some small portions in some of the exons. So now if we instead look at the results from STAR, it looks like this. So this was actually able to recover all the reads that are going across the exons. So again, each of these is a read, and if they're connected by a blue line, it means that they are spanning an exon junction. So this read starts there, and then the alignment finishes there, actually. Yeah. And so now you can see that we actually are recovering the whole gene, but you need to account for the splicing. Okay, so this is quite handy also to have. You can use the UCSC browser, is another popular to just look at your alignments. Okay. So questions on how you go from your raw sequencing rates to an alignment file. If not, then I think, oh no, we still have, yeah, we can move on. Yeah. Okay, so back to this.
Right, so the last bit, so we have an alignment file, and now we want to go from there to having counts, which is what we're going to do overall analysis on. So we want to somehow, is this still not working? Yeah.
Let's just try.
Yeah.
It's searching, so maybe something will happen eventually. Yeah.
Nope, it's the same.
Maybe just. I mean, I can also go to it and finish.
Yeah. No. Okay, so that's
Exactly. else if nothing's changed.
Yeah.
Can just- can
Just have the slides.
Yeah,
just have this.
that's probably better.
Is that's big
than
enough?
seeing the first slide.
Okay. This is the very last bit, going from alignment files to count type files. So there's multiple methods that do this as well. As we saw, star already can do that, as well as aligning. Perhaps more common are HTSeq and feature counts. You might have heard of those. What those methods take as input are the BAM file, and then some sort of features of interest we want to assign those mapping reads to. So this is just this example here on the right is from HTSeq. So let's take this to account. Feature count, it's quite similar. So the reference will be in the form of either one region, so chromosome 1, X to Y, or a union of multiple of those that often are, for example, the exons. We're mostly quantifying towards genes for this purpose, so a gene will be a collection of its exons. And then you can, so in these cartoons here, the green is the read that we have, and the purple and then also blue are the genes, are sort of a reference that we want to map or assign our reads to. And then these three are the three different settings that you can provide HTSeq in terms of whether you want to count that read in for that gene or not. So the three parameters are these ones: the union, the intersection in a strict method, and non-empty intersection. So this means that you're taking either the union or the intersection between those two, so the green and the purple. So let's just go over the examples. In the first case, it's the simplest case, the read entirely maps onto that gene, and so all three, so the union of those will be basically, in this case, the entire gene. And the intersection in both cases will be the entire read. So in both cases, the read is unambiguously assigned to that gene. So this will result in one count for this gene. The second example, the read is only mapping onto this gene, but not entirely. So there's a bit of the read that doesn't map onto the gene. So again, of course, in terms of the union, the read does belong to the gene. And similarly, the intersection is not empty because there's that bit of the read that is intersecting with the gene. So in both the first and the third parameter, we will assign this read to gene A. However,
be biologically, or could it be this example?
this
So
it's, well, I don't know,
Yeah.
many things.
any ideas?
Yeah.
No?
No?
So for example, let's say this is your gene annotation, right? So you might say that the gene goes from X to Y, but annotation is quite often incomplete. And so it might be just that the UTR hasn't been completely mapped. And so actually
Yes.
the read might be correct, but you just haven't reached the actual end of the read properly, of the gene. Yeah. So that's just a problem with the annotation in many
It's
cases.
often, yeah. It's based on what you're aligning to, the features that you provide. Okay, so I think this is quite straightforward. The third example, the read probably retain the intron in there, or the annotation has only these two exons, but really there's one in between that has just not been annotated. So again, the union will find this read to uniquely map onto this gene. Similarly, the intersection is not empty, but if we look at a strict intersection, so all of the intersection is to map onto the gene, that doesn't work. So, and if you use this very strict filter, then you won't count this read as part of the gene. In this third example, both the read and the gene are actually unions of two different bits, probably exons again. So in this case, all three agree. Then these are different examples where the same read, in this case, uniquely maps onto one gene, but it actually, at least part of it, also overlaps another gene. So if you take the union now, it's complicated. It doesn't know, because in terms of union, this could be assigned both to gene A or gene B. So it will flag this as ambiguous. the other two, obviously, because it's an intersection, will assign this read to gene A only, because it all falls within gene A. This is kind of the taking this to the extreme. If it maps onto both. Oh.
There we go.
Okay. Yeah, so maybe quickly, the last two are ambiguous because it could be well gene A or gene B. So this will be the result. It won't know what to assign it to. There's more complex programs that try to also quantify transcript abundance. So you might assign it to one transcript or another. Instead of genes, you could have as your features the transcript isoforms, for example. Some will correct biases more, so we'll be more aware of GC content and things like that. And some try to account for multimetric mapping rates, although it's not. Okay, the maybe last thing that's worth mentioning, we're not probably going to go into those, but it's worth, you should know that these things exist. So they're an alternative to the protocol that we've described so far. So mapping first and then quantifying these so-called pseudo-alignments. So the two most popular ones are Callisto and Salmon. You might have heard of them. They're actually doing very, very similar things, although they each say they do much better than the other. The idea is that you try to do both alignment and quantification in one go. This is really only for RNA. So this is really only for genes. So if we want to quantify the genes, and the idea is that it's a lot faster than normal alignments because instead of mapping the whole read, it divides the reads into k-means. You can set k. Often use 7 or some very small, and so you do not allow for mismatch. So obviously an alignment that does not allow for mismatch is only happy with perfect matches is a lot faster. And so maybe vaguely the idea is you build this k-means, then you build some sort of de Bruijn graph, so some sort of possible combinations that putting these k-means together might get, and then you assign the read, so the k-means to potentially multiple transcripts at the same time, but with different probabilities. So it is just a very quick way. So it does not work if we're trying to find different isoforms. It's really just for transcript quantification. You often give the transcript and you just count. And the way around, the memory and the length, the time is to use this very small k-means and not allow for mismatch.
So it wouldn't actually give you a full alignment. It will just look
Counts.
for
Yeah.
where it could map.
Yeah.
And then that's it. If it maps, then that becomes
The
a count.
idea is that that's enough. You don't actually give it what this is for, but just how many counts you have with this gene or transcript. So it's worth knowing that they exist. These are the papers. I think that's...
Yeah, okay. So I think it's...